Remember the BCG is a vaccination which we get for tuberculosis.
Yes.
Okay, but there is more to BCG than the tuberculosis vaccination alone. Okay, so nowadays there are certain other roles attributed to BCG vaccination. I mean, in addition to the tuberculosis prevention. Okay, so let's see. Now, it's essential to see at what level in the immune reaction these drugs act on. Okay? So, for that, you need to know first what are the stages of mounting an immune reaction. Okay? And then we'll see at what stage they work. This is a lymphocyte, pretty obvious, large purple nucleus, smaller rim or a scanty rim of pink cytoplasm. Okay, so this is a lymphocyte, typical. Recognizes the antigen, okay, with the help of antigen-presenting cell. You know it already. So once it recognizes the antigen, the immune reaction may mount. Okay, which is characterized by proliferation of the cells, the lymphocytes. So the lymphocytes, they proliferate. There's an active proliferation of the T lymphocytes. And then they differentiate depending on the immune reaction, either a B cell response or a T cell response or a mixture may mount. Okay? So either of them or a combination. So there is a differentiation into either T cell or B cell, lineage, and this may interact with the complement wherever necessary. Okay, and this complement, which is part of the innate immunity, this will lead to effective antigen binding and immune response. Okay, now immune response also includes immune injury to the native cells, because it's a battleground, so there is a collateral damage. Okay, so there is a damage to the invading organism or antigen, but at the same time there is some immune-mediated injury to the cell tissues as well. So the antigen binding and immune mounting also includes production of the chemical mediators, okay, which includes cytokines, okay, so that there is further recruitment of the lymphocytes. So it becomes a positive feedback. Okay, so this is a classical example of positive feedback where more and more lymphocyte activation takes place as long as the antigen is there. Okay. So let's see at what levels we have weapons which can take care of the uprising. Okay. So we have antigens. Antigen recognition could be dealt by antibodies. So either you block the antigen or the antigen recognizing molecules. Okay? So either you block the antigen or antigen recognizing molecules with antibodies. Then we have many of the drugs which are actually working at the proliferation phase. Okay, so corticosteroids, methotrexate, azathioprine, mycophenolate, and cyclophosphamide, for example, all of them, they work at the proliferation phase, where they inhibit the cellular induction and proliferation. And you have the immunophilin or immunophilin-related drugs, cyclosporine, tacrolimus, sirolimus. All of them, they take care of the differentiation phase, the later phase. Okay? Unfortunately, we don't have any effective drugs once the immune reaction enters the committed steps. Okay? Except some help by the corticosteroids, they inhibit and release the action. Some help by the corticosteroids, yes. But most of the times we don't have a good I would say, drug against the reactions taking place here. Okay?
said on our slide, it says that immunoglobulins are actions as immunomodulators, right? But here it says that they inhibit.
If you remember, in the very first slide, the title slide, we said immunomodulators. Immunomodulators, they are neither immunoboosters, clearly, neither immunosuppressants, clearly. I mean, it's actually a mixture of groups. Some of them, they work as an antibodies to inhibit the immune reaction. Other ones, for example, like interferon, they have a multitude of functions, but they are immunomodulators rather than the immunosuppressant or booster. Okay, so it's actually a group of compounds. That's why immunomodulator. Here we are talking about the immunosuppressant antibodies. Okay. Okay. Let's see. Now, let's start from the drug which you already know, glucocorticoid. Now, this is a very, very simplified diagram showing you that the cortisol, being a lipid, can cross the lipid bilayer of any cell membrane, okay, and enters the in the cell. Within the cytoplasm, it has its own receptor, which is a corticosteroid receptor, okay, and this corticosteroid receptor is coupled to a number of proteins or cofactors, which are then released. They have their own functions to bring about, okay? They are not there just idle. They are there for some other reasons and factors, which we are not emphasizing more at the moment. Cortisol binds to its receptor. There is a dimerization, okay? And this dimer then enters the nucleus, where it has got its own binding regions within the DNA, and those regions are called GRE, glucocorticoid response elements. Okay, good. Now the GRE, being the DNA, rather the regulatory DNA, would regulate certain genes, okay, which help in reducing the immune response. Okay? So there could be a number of ways to reduce the immune response, either give out certain immunomodulator factors like interferons, or sometimes giving out the suppressors of the immune, the interleukin producing genes, for example. So the ultimate effect is a blunted immune reaction. Okay? That's what it's doing. So, yesterday, at least in my group, we discussed, I don't know about the others, but at least we discussed the role of glucocorticoids in relationship with the beta receptors. Yeah, if you remember? We discussed something.
So we can take them out.
Okay. Okay, forget about it. So, there is an interesting interaction. Okay, a synergistic activity between corticosteroids and beta adrenergic receptors. Okay, we know that in stress response, the glucocorticoids bring about overexpression of beta receptors over various body cells, including heart and vessels, etc., etc. Okay. But there is a reason, okay? For the stress response, we need to have a good strength of the heart to pump, etc. But there is another aspect to it, is the immunomodulation. Okay? The beta receptor stimulation also helps in immunomodulation of the brought about by the glucocorticoids. Here is a diagram which shows you the glucocorticoid pathway and the beta adrenergic related function of the cytokine production activity, as well as acetylcholine. Okay, so this is an interesting interactive diagram. You can always ponder on, but I'm not emphasizing on this because it may be a little more asking from you. Okay, so interfering now. Let's see what. Remember the immunophilin-dependent drugs, cyclosporin and sirolimus and tacrolimus, we discuss it, we will see what they will be doing inside the cell, the lymphocyte. Here we are looking at that in this slide. Calcineurin and mTOR are actually the downstream, okay, molecules in the which are come across when there is activation of T cell receptor. Once T cell receptor is activated, okay, there is a whole intracellular, rather intra T cell sequence of events which involves certain proteins, okay? And the ultimate effect is T cell activation, okay, differentiation, activation, proliferation, all of them, okay. So now we are talking about that aspect. Now here is the T cell receptor, and this is the co-stimulator. You know, for effective T cell receptor stimulation, we need to have some co-stimulator. We already discussed. It's a signal. It's a pattern recognition sort of signal. Okay, so let's see what happens when T cell receptors are stimulated. The downstream immediate pathway is activation of calcineurin. Okay, it's a protein. Okay, and calcineurin depends on calcium and immunophilins for activation. Okay, immunophilin, remember. Calcium and immunophilins are required to activate calcineurin. And what are the two immunophilins which are important therapeutically? Cyclophilin and FK binding protein. Now this one and this one. These are two immunophilins, cyclophilin and FK binding proteins. And both of these are targets of two different drugs. Cyclosporin binds to cyclophilin. Okay? That's why cyclophilin, because it can bind with cyclosporin. Okay? And FK binding protein is the one which binds to the molecule which was previously known as FK506. That's why FK binding protein. Okay? Now we call it tacrolimus. Okay? The other name of tacrolimus is FK506, which we already, sorry? Yeah, it's already mentioned. So that's why tacrolimus binds to FKBP, which is the immunophilin for tacrolimus, and cyclophilin is the immunophilin for cyclosporin. Okay? So these two immunophilins, they are there to activate the calcineurin. Okay? So once the cyclosporin and tacrolimus bind to their immunophilins, the calcineurin activity is reduced. What calcineurin is actually doing, it stimulates another activating factor for the T lymphocyte, and we call it NFAT, or nuclear factor of activated T cell. Now this is a nuclear factor. That means it's a protein which translocates to ultimately to the nucleus and brings about the gene expression required for differentiation, proliferation, and active secretion of the immune-related mediators like interleukins. Okay? The ultimate effect is transcription of the lymphocyte stimulating interleukins, which is interleukin 2 primarily. Right? Interleukin 2 is a protein. So a protein, that means the nuclear region, the DNA responsible for the coding of that protein has to be expressed more. So there is a gene expression and more production of interleukin 2. Now interleukin 2, once synthesized, is released, and then it has got its own receptors over other lymphocytes as well as the self. Now once it reaches its receptor, binds to it, it stimulates its own as well as the other. So it's important in the positive feedback mechanism. Okay, interleukin 2. So it's important. The positive feedback we saw in the last slides, in the previous slides, So that positive feedback depends on the interleukin-2 receptor activation.
Silent.